So this will continue the same chapter. So what is this? This is it's called nucleoid, the circle. Okay, nucleoid. And nucleoid is circular DNA. What do you find in like eukaryotes? You don't find circular DNA. You find linear DNA, straight DNA. How many chromosomes do we have? Do you remember? How many chromosomes? Human cells. How many? Yes. Do you remember X, Y, XX chromosome? Yes, yes. How many? Rest how many? Those are sex chromosomes. 46, yeah, 23 pairs, right? So we have 23 pieces of DNA. But in bacteria, only one circular DNA. Okay, right. There's another name, it's called nucleoid. Nucleoid or circular DNA. Okay, remember that. It controls all the cell activities like protein synthesis, everything. And what's the function of nucleus in a cell? Controls the cell activity. Do bacteria have nucleus? No. So what do you call cells without nucleus? Hmm. Prokaryotes. Okay, what's the other one? Eukaryotes. Okay, right. So that's what is it? Nucleoid, right? What is nucleoid? The ring of DNA, circular ring of DNA, and it controls cell activities. And you find the second one. There's a second ring of DNA. It's called plasmid. It has extra characteristics like antibiotic resistance. What is antibiotic resistance? Do you know antibiotic resistance? No, right? No, today, if you do you know antibiotic? Like you take a medication, maybe for fever or some problem, like for 5 days or 6 days, every day at the same time. This antibiotic to kill the bacteria. Okay. Today, due to overuse of antibiotic, we cannot kill the bacteria. Bacteria has become resistant to antibiotic. It's like in India. Do you know we use mosquito repelling device? There's a liquid in that, right? And today you put that, mosquitoes are happy. They don't run away. They don't die. Why? They are resistant. They have become resistant to this chemical. So what companies do? They make stronger liquid in that. Same idea. When we keep using antibiotic, you cannot kill the bacteria because they have become resistant to it, immune to it, right? And where do you find the gene for antibiotic resistance? On the plasmid. Okay, on the plasmid. What is plasmid? It's a small ring of DNA. And what do you find on this? Extra genes. What's the extra gene? Gene for antibiotic resistance. Okay, gene for antibiotic resistance. Make sense? Okay. This is a flagellum, under electron microscope. We usually draw flagellum this way, but it's almost like you know, that's how we draw the flagellum. Can you see it's like a motor, like gear mechanism. And this is the cell membrane. This is cell membrane. And on the cell membrane, it's attached. This part is a basal body. The ring-like structures, it spins. Using energy, it spins. And this ring attached on the cell wall and cell membrane. And that's the filament, okay? So there are two parts. This is called hook and this part is called filament, right? And when it spins, the flagellum goes this way. It spins this way. That drives, okay? It keeps spinning this way. And that's what keeps the bacteria moving in the liquid. You find this only in bacteria which lives in liquid medium, like maybe blood or bacteria and water, right? So this is flagellum. What are two parts of the flagellum? Basal body. We don't learn the details, okay? Basal body controls the movement. Then there is a hook. On the hook, you find the filament. And what's the function of basal body? Controls the movement, circular movement. And that moves the bacteria. If you cut it open inside, you will find 9 microtubules. How many? Nine. 1, 2, 3, 4, 5, 6, 7, 8, 9. Okay. 9 microtubules, right? So they call it 9 microtubule. Microtubule is made up of a protein, tubulin. No, don't go into too much detail, just for understanding. So, how many no, microtubules, tubes inside, pipes inside? What's the name of the pipe? Microtubule. How many of them? Nine. So, what's the structure of flagellum? Flagellum has a basal body, a hook, and a filament. And inside the filament, inside from here, you will find this. Inside the flagellum, how many microtubules? 9 microtubules. What's the use of flagellum? Help bacteria to move. I mean, it swims in the water, right? Yeah. Describe the flagellum. Mhm. Filament, microtubules. Okay. Yes, for movement. But when we study eukaryotic flagellum, do you remember the structure of sperm in class 10? It has a tail, right? This tail is eukaryotic. So here you find 9 plus 2, 11 microtubule. Here you find 9 microtubule. So it's a difference between prokaryote and eukaryote. Okay, so describe the structure. Hmm, hmm. Microtubules. And what's the use of this? Movement, yes. Mhm. Movement, filament. What's inside the flesh? Microtubule. Okay, this is, can you see? Microtubules. We don't have to learn the details. It's like one pair. There are 9 of them. But in the middle, there's nothing. Okay, there's nothing in the middle. Right. Okay, those are the parts of the flagellum. Yeah, that's fimbriae. Can you see the fimbriae? What's the use of the fimbriae? It helps bacteria to stick to any surface, okay, or stick to each other. They stick to each other and stick to any surface using this. That's called fimbriae. Okay. Then you have ribosome. These are ribosomes, you know. Have you done ribosomes in class 10? Yes, okay. What's the function? Protein synthesis. If you look at a ribosome, there are two kinds of ribosome. In prokaryote, it's called 70S ribosome, and eukaryote, it is 80S. Okay, 70S is like a small ribosome. This is like large ribosome. Okay, 70S is made up of two subunits. You see, there are two parts in a ribosome: a large subunit and a small subunit. And large subunit is 50S, and the small subunit is 30S. Okay, but 50 plus 30 is not 70, it's 80. Then why do we say that? S stands for a unit. It's called Svedberg unit. Don't learn it. Okay, that means it shows density. Have you seen how they separate your blood cells from plasma in the laboratory in hospital? They take your blood, put it in a test tube, and spin it at high speed. They call it centrifugation. Seen or heard about it? No, right? So when you do that, the cells get separated, plasma gets separated. Similarly, they separate each piece using the spinning mechanism, centrifugation, right? And this is the density at which large subunit gets settled down in the test tube. And this is the density, and this is the density or speed at which the other one gets settled down. But when they are together, they are heavier. They come down quickly. That's why it is 70. It's not the sum of this. Okay? So, what kind of ribosomes you find in bacteria? Small ribosome. What's the other name? You can't write small ribosome. You must write 70s. 70s means small ribosome. How many subunits you find here? Two subunits. What do you call them? Large subunit and small subunit. And what's the large size of the large subunit? 50S. Okay. And S stands for Svedberg unit. If possible, learn it. Okay. Right. Otherwise, later, slowly. Right. Don't take up too much. Right. And Large ribosome is 8 years. This will study with the eukaryotic cell. So leave it now. What is it made up of? It's not surrounded by membrane. See, okay. What are the two features of prokaryotes? Sneha, no nucleus. No, excuse me. Cell membrane is there. Say another feature. Cell wall is there. Bacteria has cell wall, peptidoglycan. Say another feature. Membrane bound organelle. What's that? It's not covered by membrane. Okay. So this ribosome is not covered by a membrane. Keep that in mind. And what is it made up of? Ribosomes are made up of rRNA. Do you know RNA? Do you know DNA? Yes. Okay. I think you studied protein synthesis. Yes. DNA forms mRNA. No. Okay. Anyway, RNA is another type of molecule produced from the DNA. Okay. But we'll study it in all these things are in different chapters. But here you just learn the name, only the name, no details. Okay. What is it? RNA. Have you studied about viruses? Virus structure. That's class 9, right? The classification. You did virus. Anyway, okay. Okay, so that's RNA. What is RNA? It's a type of genetic material, taken this way, type of genetic material, right? And this is made up of rRNA. Or just learn RNA and protein. So what is it made up of? RNA and protein. There's no membrane around it. And it has two subunits, right? Two subunits. And what is its function? The function is protein synthesis. Protein synthesis. Okay. Now, this are the sizes. I don't force you to learn it now. We need it for the NEET exam, but later, slowly we can learn, right? Okay. What is 1 nanometer? 10 to the power 9. Okay. What's the other one? Micrometer, right? Micrometer. What's the second one? Nanometer. Okay. What is micrometer? 10 to the power 6. Nanometer? 10 to the power 9. Physics, they'll teach you. But doing it now, right? In the beginning, they teach you all those things. Learn everything really well. Physics is a curse on bio students. Are you math bio? Only bio. Okay. This is the biggest trouble for bio students because you study engineering physics. We don't need this. Doctors don't need engineering physics, but they teach you that. So from the very beginning, the sir must be teaching you, like your teacher, the the fundamentals, like units and conversion, just by heart. Paste it on your wall, in your toilet, everywhere. Okay, and keep looking at it. It should be like ABCD of nursery. Then it become easy. Okay, so take care. Anyway, so what are two things? Micrometer, nanometer. Nanometer is smaller than micrometer. Okay, fine. Yeah. So this is it. So what's the structure of ribosome? In bacteria, you find what kind of ribosome? 70s. Ribosome, in ribosome, no, it's not membrane bound. It's not membrane bound. That means it's not surrounded by any membrane. Both. So that's a similarity, between eukaryote and prokaryote. Both have ribosomes, and they're not surrounded by membrane. Okay, and what is this made up of? RNA and protein. Just RNA and protein. It's made up of RNA and protein. And there are, what's the size of this? It's called 70s. It's made up of 50s, large subunit, 30s, small subunit. Okay. And function, what's the function? Protein synthesis. Yeah, try. The ribosomes of bacteria, prokaryote. Yes. 70s, small subunit, 30s. Synthesis. Yes. Okay. Is it surrounded by a membrane? No. Okay. Try. Bacteria. Ribosome. 70 years. 50 years. 50 years. Mm. Is there any covering around it? No. Okay. And what's the function? And what is it made up of? And protein. Yeah. Try. 30 years. Yes. Yes. Protein. Okay. Right. There you can see it. It's performing. This is protein coming out. Right. This will study in class 12. This is how protein is being formed. Okay. The green line is mRNA, and those are the protein, polypeptide. Okay. Don't learn this part now. Okay. There are many substances in the bacteria which are not actually, all the bacteria. They call them inclusion bodies. Okay. What's inclusion bodies? You will find some of the some of them have gas vacuole. Blue-green algae, blue-green cyanobacteria. Studied? No. Nine. Yeah, heard of it, right? Heard of blue-green algae, cyanobacteria, nitrogen fixation in the root legumes? No. Okay. Anyway, so in some bacteria, you will find gas vacuole. It helps them to float. It's called inclusion body. It's an extra thing. Okay. Sometimes you find glycogen. Where do you find glycogen? In animal cells. It's a stored food. It's like starch. Where do you find starch? In potato. What instead of starch, what do you find in animal cells? Glycogen. Okay. Same thing you find here. Glycogen you can find here, right? And these are known as inclusion bodies. Okay. So what are inclusion bodies? These are the extra substances you might find, like glycogen, gas vacuole, are known as inclusion bodies. Is it good? Okay. Yeah. So we finished prokaryote. All right. So prokaryote. What's a prokaryote? Hmm. Nucleus and membrane bound organelle. Okay. What kind of ribosome you find? 70th ribosome. What are subunits of 70th ribosome? Yeah. What's a cell wall made up of? Glycan. Okay. What uh, structure is used for sexual reproduction by bacillus? And how does it attach itself to other surfaces? And that's pilus. You're confused in that. Pilus for yeah, attaching. Fimbriae. Fimbriae, right? Name the small ring of DNA. Small ring of DNA in the bacteria. Plasmid, right? Okay. What do you find on plasmid? What's a special characteristics gene you find on plasmid? Something related to antibiotic? Antibiotic resistance gene. Antibiotic resistance gene. Do they have a nucleus? Then what controls cell activities? Nucleoid. What is it? Nucleoid. What's nucleoid? A ring of DNA. Nucleoid. What's nucleoid? Nucleoid is just a ring of DNA, right? Ring of DNA. Okay, we did this. It's nucleoid, a circular ring of DNA. What's a small ring of DNA? Plasmid. What do you find on plasmid? Antibiotic resistant genes. Okay. And what is mesosome? Mesosome. Infolding of. Why is it folded inside? Say one advantage of this. No. DNA. No. If it's folded, what's the advantage? Increases surface area. Okay. Say one function. Yeah. Two opposite side. Okay, one more function. Respiration, enzyme for respiration. Anything else you remember? Separate the chrome nucleoid, DNA to both the sides, right? Okay. So you're not bad. It's good. You remember things, okay? But the volume keeps increasing very rapidly. In school, they just keep flying. In school, teacher has only one aim to finish the syllabus. The kid just keep rushing, and you get stressed out. So keep studying. If you keep studying, your information keeps getting collected. You become more wise in the subject. But if you don't revise, focus mainly on tuitions. In 11th, don't expect anything from school. School, you go for fun. Okay? Your school is better. But ICSK is just hopeless, right? There they want to finish. Here, at least they try to teach something, right? So focus more on tuition, and almost all tuitions will go with the school together, right? Yeah. And let's do eukaryotic cell. Tell me the features of eukaryote. One feature. Why do membrane bound organelle? Another, there's a nucleus. Say something else. Anything else? You studied. Now, do they have a different ribosome? ATS, ATS ribosome. We'll do it now. Okay. So these are the features: membrane bound organelle and nucleus. Okay. They also have chromosomes. We'll come to that. So these are the features. Look at the cell membrane, cell wall for eukaryotic plant cell. What makes plant cell different? Plant cells have cell wall. What is cell wall made up of? Cellulose. Okay. This will study endomembrane system. Have you done any of this? ER, you did? Golgi, you did? Okay. Lysosome? Okay. So familiar. Mitochondria, you did, right? Plastids, ribosomes, which is ATS, cytoskeleton, the skeleton inside the cytoplasm. It's called cytoskeleton. Then cilia and flagella. Where do you find cilia? Like we have cilia in the trachea, okay, to move the mucus up, right? Flagella you find in sperms. What's the difference between sperm flagella and bacterial flagellum? Bacterial flagellum is prokaryotic. So it has 9 microtubule. But sperm is eukaryote, 11 microtubule. And this is for, you know, forming spindle fibers. Do you remember mitosis? Mitosis, meiosis, right? Okay, so we'll study that. Then there is a nucleus and microbodies. Okay, and this for animal cells. Animal cells, you don't find. Okay, this centrosome, this is absent. Cell wall is absent. Okay, in animal cell, there's no cell wall. There's no chloroplast. Okay, no vacuole. Don't memorize it now. Let's study about cell membrane. And cell membrane of bacteria and cell membrane of eukaryotes are the same. No difference. Okay. What is cell membrane made up of? It's made up of three things. Phospholipid. Okay. By the way, we have a chapter on cell membrane. Okay. Nice. Something. So no need to worry. Phospholipid, and it has protein, and it has cholesterol. So what are three things? Three things in cell membrane. Cell membrane is made up of three molecules. What are three molecules? Phospholipid, protein, and cholesterol. Keep learning. The key part of your study is memorizing. Okay? In biology, I'll make you memorize in the class. That's the best way to study. Then you go home, you feel, oh, everything is easy. Otherwise, you have to sit down and repeat, then you don't understand. Okay? So when I explain something, say something, you keep saying in your mind, repeat. Repetition is the key. It will stick. Don't worry. After some time, it becomes so easy for you. You just listen and it registers in your mind. Okay? Right. So what is cell membrane made up of? Three molecules: phospholipid, protein, and cholesterol. Okay. phospholipid, protein, and cholesterol. Cell membrane of bacteria, cell membrane of eukaryotes have the same molecule. So no difference. Okay. So these are three molecules. We'll study about each of this. But I'll explain about phospholipid. Yes. Okay. That's phospholipid. This is a one phospholipid molecule. Okay. One phospholipid molecule. It has head and two tails. Okay. What do you find here? Head and tail. Okay. We'll animate it so it looks nice, Okay. Head and tail. What's special about the head likes water. Okay. So it keeps sticking to the water. Hmm? Water. Yeah. So it's called hydrophilic. Means it likes water. Head likes water, right? But the tails hate water. Okay, it's called hydrophobic. This you did in chemistry? Yes. So it's called hydrophobic. Understood? So what is phospholipid made up of? Two parts. Head and the tail. What is head? What's special about head? It likes water. What do you call if it likes water? Hydrophilic. And the tails hate water. What do you call the tail? Hydrophobic. Okay. And cell membrane has two layers of phospholipid. So this is one layer. These are the tails. Do you see that? See, two layers. And what do you call this? Phospholipid bilayer. What is it? Phospholipid bilayer. Name the three molecules of cell membrane. What say the structure? Describe the structure of phospholipid. Tail. Philic. Hydrophobic. How many layers of phospholipids you find? Two. So what do you call this? Phospholipid bi. Bi means two, right? Bilayer, right? Okay. Say name the molecules of the cell membrane. Cholesterol. What is describe phospholipid? Head. Tails. What's special about head? Mm-hmm. Phobic. And how many? What do you call the cell membrane? The phospholipid of bilayer. Okay. Molecules of the cell membrane. Mm. And what's the structure of phospholipid? Hmm. Yes. Hmm. Hydrophobic. And when they come together, what do you call the layer? Phosphobilayer. Okay. Now look at this. This is a cell, right? If that's a cell, what do you find inside? Cytoplasm. What do you find outside? There is a liquid known as tissue fluid. Have you heard about this in tissue fluid, right? Okay. Means it's like water from the plasma of the blood. So inside cytoplasm, what is cytoplasm made up of? Mostly water. And what is tissue fluid made up of? Mostly water. Okay. So you're looking here on a piece of cell membrane. Okay. So if I draw this way, this is a complete cell membrane. See, this is a cell like that. Okay. So if that's a cell, what comes here? Cytoplasm. And what is here? Tissue fluid. Okay. I'll take this part. Let's say it's this piece. And that's a whole cell membrane, cytoplasm inside. Make sense? Tissue fluid outside. Can you see head of the phospholipid is facing cytoplasm? What's the reason? Why is it facing the cytoplasm? The head is facing there is water inside. So what do you call the head? Hydrophilic. Okay. And why this head is facing the tissue fluid? Why is it contains water? Okay. So hydrophilic heads of both the layers face either cytoplasm or tissue fluid. And where do you find hydrophobic tails? Away from water. Okay, it's away from water, right? This prevents water from entering. Water cannot freely pass through it because of this tail. Tail repels water, stop the water from entering. Make sense? You have a question? Do you have a question? Yeah. So how many layers of phospholipids and how they are oriented, organized? Heads of both the layers are facing either tissue fluid or water. Cytoplasm. Where do you find the tails? Between the layer. What do you call the layer? Phospholipid bilayer. So what is cell membrane made up of? Phospholipid, protein, cholesterol. Okay, three things. And what are the parts of the cholesterol? Phospholipid, head and tail. What is special about head and tail? Hydrophobic. What do you find? How many layers of phospholipid in a cell membrane? Two. What do you call that? Phospholipid bilayer. Okay. How they are organized? Head is facing cytoplasm or tissue fluid, and tails are between the two, away from the water. What's the advantage of having hydrophobic tails? It stops the water from freely moving through it. What if water simply goes in? The cell will burst. So this tail stops the water from freely moving in and out. Is it good? So that's phospholipid. There you can see, right? This phospholipid bilayer. What keeps a phospholipid together? The phospholipids are held together by a molecule known as cholesterol. Okay? Even cholesterol, you find two parts. This is the cholesterol, but one tail. Cholesterol has hydrophilic head. That's why it's stick to hydrophilic head. And its tail is hydrophobic. Okay? Both have the same feature, but different structure, right? The tail is what is tail? Hydrophobic. Hydrophobic. Tail is hydrophobic, head is hydrophilic. What's the function of cholesterol? Keep the phospholipids together. Hold the phospholipids together. So how many molecules in cell membrane? Three. Name them. Yeah, cholesterol. And what's the structure of phospholipid? Head and two tails. What's the structure of cholesterol? One head and one tail. Okay. Which part of phospholipid is hydrophilic? Head. Which part of cholesterol is hydrophilic? Head. And tail is hydrophobic. And where do you find the cholesterol? Between phospholipids. And what does it do? Keeps the phospholipid together. Hold the phospholipid together so that they don't break up. Right? Okay. Then you find proteins. There are two kinds of proteins. One protein is it spans through the membrane. Can you see it's throughout the both the layers. What's the name of this part? What is A? Phospholipid bilayer. What is B? Head of the it means a phospholipid. Okay. And what could be this one? See? Yeah. And can you see this protein spans whole membrane? So they call this integral protein. Okay. Integral protein. It's inside the membrane. And this is either only on one side. See? Either inside or outside. So this is called peripheral protein. So what are two kinds of proteins? Integral protein and peripheral protein. So what are three components of the cell membrane? Phospholipid, protein, cholesterol. How many types of protein? Two types. What are two types? Integral protein and peripheral protein. What's the difference? Integral protein throughout the membrane and peripheral protein only on one side. Only on one side. Okay. So these are the types of protein. What other molecules you find in membrane? Two layers of what? Phospholipid. So what do you call that? Phospholipid bilayer. What keeps phospholipids together? Cholesterol. Where do you find hydrophobic part of phospholipid? Between the two layers. Hydrophobic, right? And why is it there? Water it stops the water from moving inside. Okay, is it good? Name the two types of protein. Mhm. Peripheral protein. Okay, now look at the function of these protein. And these are integral protein. What do they do? You know, through these proteins, some of them transport happens. Okay, what kind of transport? It's in another chapter, but is related to that chapter. Through this membrane, do you remember osmosis, diffusion, active transport? Active transport is moving molecules from low to high concentration. Okay, yeah. Anyway, so this is passive transport, means diffusion. Through these through these molecules, diffusion happens. Okay, by diffusion, molecules enter the cell. Right. These things are in the syllabus, but I'm not explaining it now. It's in another chapter, right? And this is called active transport. Active transport molecules move from low to high using energy. Are you familiar with ATP? Okay, ATP is energy, right? Low to high. But here they are moving from high to low. So what happens through what's the function of these integral protein? Transport. Okay. What are the two kinds of transport? Diffusion and active transport. Okay, there's another kind of diffusion. Don't learn now, later. Diffusion and active transport. What is active transport? When molecules move from low to high. What is diffusion? Molecules move from high to low. Is it good? Okay. And the membrane has a name. Okay. It's called fluid mosaic layer. Okay. So it's also known as fluid mosaic model. Okay. Two scientists, Singer and Nicholson. These names are very important. Okay. Very important for NEET exam they ask. Even school exam for 1 mark they ask. Multiple choice. They called it fluid mosaic. Have you seen mosaic on the floor in old villas buildings? On the floor you find tiles with a lot of spots. No. Have you seen mosaic artwork on the walls? Mosaic. Do you know tobacco mosaic disease? No. Okay. So that means I'll show you what is mosaic is. What is mosaic? Yeah, this kind of art. You see that? This is like mosaic art. See, they put pieces of. Have you seen this kind of art? Like in see, this is like art mosaic, right? Have you done this kind of thing? Like you might have done you tear the paper, different colors, and stick and create pictures. Yeah, this is called mosaic, right? Okay. So what's the name of the membrane model? Is fluid mosaic model. Okay. Imagine you look from the top. Okay, you're looking down at the membrane. You see uh, phospholipid heads, lot of phospholipid heads, and on that you find proteins scattered. Is it like a mosaic? Okay, in the background of the fluid mosaic, I mean fluid on the background of phospholipid heads, you find scattered proteins. So that gives appearance of mosaic. But the other mosaic we see is permanent. It won't move. This moves. These proteins are floating like icebergs, ice cubes in the water. It keeps moving all the time. So they call it fluid mosaic model. Why is it fluid? Because proteins move. And why is it mosaic? Proteins are scattered. So that gives you appearance of the mosaic, right? So it's called fluid mosaic model. What is this? What kind of protein is this? Integral. And this is integral. But can you see? on the membrane. These are peripheral proteins. You will find uh, carbohydrate on it. Then they call it glycoprotein. You studied about blood groups. What is blood group? Glycoprotein. So blood groups are glycoproteins sticking out on the red blood cells. Can you take blood from a different person who is not matching with you? No. You die, right? Because the glycoproteins do not match. So that's a function of the peripheral proteins. Most of the peripheral proteins have function like this. Peripheral proteins may be glycoproteins. Glycoproteins function as antigens, like blood group. Antigen? Antigen A, B, okay, or blood group. Just call it blood group. Okay, second part. Uh, some of them, peripheral proteins are like enzymes, function as enzymes. These are examples of peripheral protein. What's the function of integral protein? What do integral proteins do? Transport. Okay, integral proteins perform diffusion and active transport. Diffusion and active transport. So, what are two kinds of protein? Integral protein and peripheral protein. What's the function of integral protein? Transport, right? Diffusion and active transport. Peripheral protein. Some of them function as enzymes. Some of them function as antigens. Example, glycoprotein. Glycogen, glucose. Okay, glyco means carbohydrate. Protein with carbohydrate. So glycoprotein. No, both. For active transport, there should be a pathway, right? How the molecule goes in through an opening, right? So this is done by integral protein. Good. So let's summarize cell membrane. What is cell membrane made up of? Phospholipid, protein, and cholesterol. And what's the function of cholesterol? Keep phospholipids together. Okay. What are two kinds of protein? Integral and peripheral. And what's the function of peripheral protein? Like some example like glycoprotein for act as antigens and some function as enzymes. Okay, integral protein, what's the function? Yeah, diffusion and active transfer. Good? Okay. Yeah, there we can see cell membrane, right? Phospholipid bilayer. What kind of protein is it? Integral protein. This is a carbohydrate sticking out. Okay. This is integral protein. For what? Diffusion, active transport, right? Hmm. Glycoprotein. A protein has like this one. This is protein, but it has carbohydrates sticking on it. It's called glycoprotein. If it's phospholipid with carbohydrate, they call it glycolipids. Okay, glyco means carbohydrate. Okay. These are two people, right? Singer and Nicholson, right? That's a function like transport of molecules, passive and active transport. Okay, this part we don't have to learn now. It's in a different chapter. Take a break. Then we'll study cell wall, right? Okay. 5 minutes. Okay, good. Try to recall what is studying, like cell membrane, what is made up of. After studying, we just forget. Even after tuition, you try to recollect. That means you're enforcing, reinforcing your memory. We don't want to think about study. But now change this, and you see it becomes so easy. Even after the tuition, try to reflect. What is it? What is it made of? Ask yourself. Try to bring it back. So that's revision happening in your brain. You don't do anything. You just keep thinking. And then you feel that, oh, I forgot this. Check. Again, keep thinking. And this way, you can be very good in class 11, even me. These are basic. Make it very difficult. But if you score like 400 plus, you get a C. No, without getting any damage. 400, 450, within management quota and NRI quota. Merit is like, it goes up. Out of 700, we need 650. About 600. It's achievable, right? But you have to force yourself, focus yourself, and always remain connected with the subject. That should run in your brain all the time. Then it makes it. This year, don't go for any activity. In school, especially in university, they force you to do everything. And then everyone fails. Last in the world. In this school, most students fail. They make them fail. And then they push them into there's another syllabus there. Yeah. And then school gets more money. And they don't teach in school all kinds of activities, and the blame is on the student. They say you did study. So don't get into any of the trap. They're giving activity, less friendship, you can have it, but you know the limits. And reinsert a subject, all the three subjects. And you find this is the most important year in life. If you do well this year, then you'll get into medicine. You keep going. Keep going. Okay? And after the school, what decide is this year. Okay? If you don't get into medicine, there are many options. You can go for any of those options. But keep medicine or dentistry number one spot. Because that's something you get good job. Even if you don't join anywhere, often a clinic, people come. In India, it's full of disease, everyone is sick. So you keep getting a lot of money. Other courses, you have to depend on someone, like a hospital, some other company. Be a doctor, really help you to survive on field. Can we start? So what is plant cells made of? Plant cells have cell wall, right? What's the cell wall made up of? Cellulose. Okay, look at this. Let's say this is a plant cell. See, that's a plant cell. Next to this plant cell, you will find another plant cell. See, the cells are stuck to each other. See, these are the cells. Let's call this cell A. Cell A is surrounded by 1, 2, 3, 4, 5, 6 cells. Like this room has another room there. But between the two rooms, there's a common wall. There's no wall for this room and that room. No. Isn't it? Same idea. Between these two cells, there is a common cell wall. This common cell wall is called middle lamella. Lamella. Yesterday we studied lamella. Where? Which part of bacteria? Yes. Where was it? Hmm. Mesosome, right? Lamella. These words keep repeating in all the topics. Okay. And now you find it difficult. Once you learn, later many things are repeated. Okay. Lamella. It's called middle layer or lamella, right? Middle lamella. Middle lamella is made up of pectin. Heard about pectin? No. Okay. It's a chemical called pectin. Okay. Calcium pectate. That's a middle lamella. So what's a common cell wall between two cells? Middle lamella. It's like a glue. It's like a glue. It keeps cells stuck to each other. Okay. So it's a common cell wall. And inside this, you find another cell wall. This is that cell wall of A. And this is cell wall of 1. Okay. So this is not common. What is common between A and 1? Middle lamella. That's common. That keeps them together. But this has its own cell wall. This cell wall is called the primary cell wall. What is this? Primary cell wall. What is primary cell wall made up of? Cellulose. Okay? Primary cell wall is made up of cellulose. See. But if the cell wall is this way, each cell become isolated. It become like a prison. What happens in the prison? People are in different rooms, but they don't talk to each other. No communication. It's not allowed. Same thing can happen. But how does it function like a tissue? Do you know what's a tissue? A group of cells functioning together. So means cells have to talk to each other. To talk to each other, cells have like channels for communication. See, these are openings on it. Okay, and this is called, these are openings between the cell. They call this plasmodesmata. Okay, plasmodesmata. What is it? Plasmodesmata. Okay, I know it's hard name, right? Plasmodesmata. No, plasmo in cytoplasm. So the cytoplasm goes from one cell to the other. Molecules keep moving to each other. It's like a hole on your, you in your room. You have a hole. Through that, you tell your mom, "Please bring tea." Something like that. Okay. So this cell is talking to this cell and this cell through these openings. What is it called? Plasmodesmata, right? So what's the common cell wall? Middle lamella. And uh, what's the other cell wall? Primary cell wall. There's more, but in a different chapter. Okay. So time being, learn this one. What's the opening between them? Plasmodesmata. What's the use of plasmodesmata? Cell to cell communication and transport of material. Okay. So what's a cell wall? Yeah, look at this. This is, okay, when you go back. And what could be inside this? What do you find below this? Cell membrane, right? There is cell membrane here. And then there is vacuole, cytoplasm, everything. So that's middle lamella, primary cell wall, cell membrane. Okay. Can you see this? That's a cell membrane. This is a primary cell wall, middle lamella. Middle lamella, primary cell wall, cell membrane. Make sense? Those are cellulose fibers. It is, cellulose fibers, hemicellulose, many things are there. But basically it's made up of cellulose, right? Okay. So what is cell wall made up of? Yeah, it's a common cell wall. Middle lamella. What is middle lamella made up of? Pectin. Okay, pectin. And what's the uh, next layer? Say a primary cell wall made up of cellulose. And what's below that? Cell membrane. Okay. Now, these are like variations. It's in this chapter. This you have to learn. If you look at algae you find cellulose and galactans, galactose, lactose. It's a sugar. It's a form of sugar. And this is mannans, okay? And minerals like calcium carbonate. In algae, you find many other things. But the other plants, what do you find? Cellulose, only cellulose, okay? These are exceptions. So what are the substances in algae? Cellulose, galactans, and mannans, mineral calcium carbonate. Okay, you try this learning. So other than cellulose, galactans, galax galaxy, all right, galaxy, galactans, right? Mannans, mannans, and calcium carbonate in algae, right? And cellulose is a main component, but you'll also find hemicellulose between the cellulose fiber, pectin in the middle lamella, so sometimes protein. Okay, what is primary cell wall made up of? Cellulose only. But this is from another chapter. There could be a secondary cell wall made up of substance called lignin. Lignin. Okay. Okay. So what's a cell wall? In cell wall you find middle lamella. What's the next layer? Primary cell wall. What's the next layer? Secondary. Okay. Secondary you don't find in every cell, some cell. Secondary cell wall, right? And if it's algae, what do you find in algae? Cellulose, galactans, mannans, calcium carbonate. Good. What is middle lamella made up of? Pectin. And the primary cell wall? Cellulose. Okay. Cellulose may contain other things like hemicellulose, pectin also. Secondary cell wall mostly lignin. Do you remember xylem? What is xylem made up of? Lignin. Xylem is made up of lignin. Okay. So what is secondary cell wall made up of? Lignin. Primary cell wall mainly cellulose. At least learn cellulose. Other things we'll learn together, like hemicellulose later. But basically cellulose. Middle lamella, pectin. Okay. What is algal cell wall? What else you find in algal cell wall? Galactans, mannans, and calcium carbonate. It's good, right? Yes. Yeah, that's what. Yeah. Why there is a cell wall? What's the function of the cell wall? Give shape to the cell and prevent the cell from bursting. What's the function of cell wall in bacteria? Same function. Give shape. Name the shapes of bacteria. One shape. Say one shape. Hmm. Bacillus, what shape? Rod. Another shape. Hmm. Round. Another one. Hmm. Spiral. What shape? Can you try one more? Vibrio. What is it? Vibrio. Comma shape. Comma shape. Okay. So same thing. Here, cell get its shape from what? The cell wall. Prevents bursting of the cell. Helps cell to communicate with other cell. Name the channel for communication between cell. Plasmodesmata. What is it? Plasmodesmata. Okay. For communication. Okay. Right. Can we do one more? Or it'll be too much? Yeah? Okay. It's all related, right? The first one, this is called endomembrane system. Inside the cell, there is a system of membrane. It's called endo, means inside. Membrane system. What are three parts? Endoplasmic reticulum, Golgi complex, lysosome, vacuole. Learn this one. You're familiar with all of them, right? Okay. Name the parts of endomembrane system, endoplasmic reticulum, Golgi complex, lysosome, and vacuole. Okay? Yeah, say. Mhm. Golgi complex, yeah, vacuole, reticulum, complex. Try. Mm. Apparatus. What's a channel for communication between cells? More does matter. What is it? Yeah, matter. Okay. This is the nucleus. And around the nucleus, you find folded up membranes. What is this? Endoplasmic reticulum. And on this endoplasmic reticulum, you see ribosomes. As what's the function of ribosome? Protein synthesis. And here protein is produced and protein goes inside, the space inside, the lumen, and protein gets folded up inside. Okay. And this is called rough endoplasmic reticulum. Can you see it looks rough because of ribosome, right? So what is RER? Rough endoplasmic reticulum. What do you find on rough endoplasmic reticulum? Ribosomes. It forms protein. Protein goes into the space inside the endoplasmic reticulum, get folded up. Okay, that's one type. The second type, look at this part. Do you find any ribosomes? No. So this is called smooth endoplasmic reticulum. And the function of this, synthesis of fat or lipid. So what are the two types of endoplasmic reticulum? Rough and smooth. Okay, rough and smooth. What is rough endoplasmic reticulum? Yeah, contains ribosomes. And what's the main function of RER? Protein synthesis. And what's the other one? SER. SER. And what is SER? Smooth endoplasmic reticulum. And what is its function? synthesis of fat. What's a channel for communication between plant cell? Plasmodesmator. Very good. Okay. And see, there's a space inside. What It's called, they call it lumen. Learn the lumen now, right? What's the space inside? Lumen. Inside the lumen, what happens? Protein gets folded up. Folding up of protein happens inside the lumen of endoplasmic reticulum. So name the types of endoplasmic reticulum and smooth. Yeah. What's the function of RER? Hmm. Mm-hmm. Yes. Okay. And the smooth one, what's the function? Synthesis. Synthesis means formation of fat. Types of endoplasmic reticulum. What? Smooth. What do you call space inside the lumen, right? And function of both? Mm. Yeah. Hmm. Fat. Yeah. Types of. Mhm. What's the space inside? And what's the difference in the function? Mm. Protein. Fat. Okay. Right. This is called Golgi. Okay. Some people call it Golgi. Okay. It's up. No problem. Golgi body, Golgi apparatus. Okay. Golgi complex are different names. And can you see here? These flattened structures are known as cisternae. Okay. Even here, these flattened structures have a name. These are cisternae. But the cisternae are interconnected. Can you see? It's continuous. But here, see the difference? They are not connected. They are not interconnected. They are parallel to each other, cisternae. Plus, can you see change in the length? This is small and this is big. Again becomes small. So it's not the same dimension. It change in the size, right? And they also produce round structures known as vesicle. Those are vesicles. Okay. So what do you find here? Vesicles and cisternae. And the space inside. What's the space inside? Lumen, like the previous one. Okay. Even here you find this. Even they can produce vesicles. Right? Even this has cisternae. But what's the difference? They are interconnected. Interconnected. And what happens here? In the rough endoplasmic reticulum, ribosomes forms protein and forms the protein get folded up inside the lumen. And this is packed into vesicle. See? This vesicle is coming from where? From the RER. The vesicle is coming from RER. And it fuses with the Golgi. Okay? It fuses with the Golgi and the protein enters here. Once the protein enters here, protein gets modified. Protein passes through this. Protein gets modified. So modification of protein happens in Golgi. And then it gets packed here and it's released outside the cell. It's called secretory vesicle, right? So it's a modification of protein. Understood this? So in RER, what happens? Protein synthesis. And this protein get folded up in the lumen of RER. Then it's packed into this transport vesicle. It fuses with Golgi. And inside the Golgi, protein gets modified and it comes out as secretory vesicle and goes outside the cell. Like we have enzymes in the saliva. Where does it come from? Salivary gland. And the cells of salivary gland is producing all the time enzyme and it keeps releasing into a tube. The tube carries it to your mouth. Okay, the same thing is this is happening in your salivary gland. Does it make sense? So what's the function of what is Golgi made up of? Cisternae and vesicle. What's the space inside? Lumen. And what's the function here? It's actually receiving protein, modify the protein, and release protein in secretory vesicle. Okay, and this forms lysosomes. Lysosomes are produced by Golgi. Okay, that's another function. Okay, lysosome is produced by Golgi, right? And this phase has a name. It's called cis phase. What is cis phase? Where vesicles fuse with the Golgi. What is this called? Trans phase. Is it good? Okay, try. What is Golgi made up of? Cisternae and vesicles. Okay. And what's the space inside the cisternae? Lumen. Okay. And uh, where does the protein come from? Incoming vesicle. Where is it coming from? RER. What happens in RER? Synthesis. Folded. Okay. And it's sent from there. It's coming from there. It fuses with this. And inside the Golgi, what happens to protein? Modification happens and packed into secretory vesicle on which phase? Transphase and released outside. What's another function of this? Lysosome. Can you say everything? Structure. Say the structure of Golgi. Mhm. Yes. What's unique about the cisternae here? Yeah. So not in the same shape, not interconnected, but not parallel to each. Yes. Space inside. What's the space inside? Lumen. Okay. Yeah. Yeah. Protein is folded in area in the lumen, and it comes here through incoming transport vesicle. Mm. Yes, yes. It's packed in what? Vesicle and released outside when the transphase. Good. So another function of Golgi, lysosome. Yeah, try. Say the structure first. Mm. Vesicles. What is special about cisternae? Same length. Okay, same size. Okay. What else? Are they interconnected? No. Okay. And what does it receive? From where? Endoplasmic reticulum. Okay. What happens to protein inside? Protein. What happens to protein? Protein gets modified, and then it is packed into. Ah no, packed into secretory vesicle. On which phase? Trans phase. What's the other phase? Cis phase. What's say another function of this? Formation of lysosomes. Is it good? Okay. So Golgi is made up of two parts. What are two parts? Cisternae and vesicle. And these cisternae have a space inside. What do you call it? Lumen. Are they parallel to each other? Yes, they are parallel to each other. Okay. But they are not interconnected like ER, left endoplasmic reticulum. And where does it receive the protein? From the RER. This protein is modified inside the Golgi. And then it is packed into secretory vesicle and released outside. Okay. Yeah, there's a function. That's lysosome. Lysosome, what forms a lysosome? Golgi. Golgi forms lysosome. Okay. And lysosome has one membrane. How many membranes? Don't know. ഉണ്ട് ഇവിടുത്തെ ആ ആ ഉണ്ട് ഉണ്ട് പോക്കോ പോക്കോ ആ so it has one membrane and what's inside? Hydrolytic enzymes. Means digestive enzyme. It's filled with digestive enzyme. Okay. So what are the two parts? Membrane. Do you remember this is called suicidal bags? Yeah, suicidal bags. And it digests the bacteria that get inside. It digests dead parts of the cell. Okay. Class 10 or 9? 9. Okay. You remember it. Good, good. Right? How many membranes around? How many membranes around ribosome? One? No. How many membranes? Ribosome. How many? None. There's no membrane around ribosome. How many membranes in Golgi? One. How many membrane in endoplasmic reticulum? One. Okay? We didn't study anything else. All of them have one membrane, but ribosomes, no membrane. Okay, if they ask you, name an organelle without membrane covering, ribosome. Okay, right. And what are the enzymes here? All kinds of digestive enzymes. Lipase, what does lipase do? Digest fat. Do you know lipid? Lipid means protease digest protein. Carbohydrates digest carbohydrates. So all molecules are digested by these enzymes. They call them hydrolases. What is hydrolase? Digestive enzymes are known as hydrolases. Okay. And what's the pH inside? It has acidic pH inside. Okay. To keep the enzymes active. Yeah. Let's summarize this, right? What is this? Lysosome. What forms a lysosome? Golgi. Okay. And how many membrane around it? One. What do you find inside? The hydrolases means digestive enzymes. Hydrolases means digestive enzymes. Can you give examples? Lipase, carbohydrates, protease. Okay. Protease, carbohydrate digesting enzyme, and lipase. So this is called lysosome. Good? Okay. And that's a vacuole. Where do you find vacuole? Large permanent vacuole in plant cells. Plant cells you find large permanent vacuole. And it has a membrane around it. What's the name of the membrane? Tonoplast. And the liquid is cell sap. Okay? So vacuole, how many membranes? One. Ribosomes, how many membranes? None. Okay? All the organelle we studied only have one membrane till now. What's the liquid inside? What do you call the liquid? Cell sap. What is cell sap made up of? Glucose, amino acids, water, and waste product of the plant. Okay? You get drugs, even coffee. Coffee is actually stored, the chemical is inside the vacuole of the cell, right? And this is called cell sap. Name the membrane here, tonoplast. This membrane has a special name. What's tonoplast? The membrane of vacuole. What is plasmodesmata? Between the two cells. What is middle lamella? Cell wall between two cells. And where do you find cellulose? In which cell wall? Which cell wall? You find primary cell wall, right? And what do you find on secondary cell wall? Lignin. Lignin. Okay. So, what's the membrane of vacuole? Tonoplast. Tonoplast. What's the liquid inside? Cell sap. What is cell sap made up of? Water, glucose, amino acid, and excretory products. Okay, excretory products. So this is vacuole. What's the function of vacuole? It keeps the cell. It keeps the cell turgid. Do you remember turgid? Like filled up with water. Leaf, fresh leaf. It's turgid, so it's upright. If it loses turgidity, it become floppy, right? So this is turgid, like a balloon filled with air or cell filled with water, like turgid. Okay, that's turgid. All right, that's enough. Enough for today. We'll do it next lesson because she's getting very restless. Yes. Yeah. Too much information, right? Yes. Slowly you get used to it and buy a notebook for this kind of notebook for test and to write question answers. I send you questions, right? Yesterday. Print them out and put notes, questions all in one folder and one book for writing. Okay, start answering from now. Look through that. If you don't know something, skip them. We'll discuss here. But the ones you know, keep answering. Leave space. Like don't write in same sequence, leave space and go ahead. And we'll have small test every lesson. So tomorrow what time? 10 is okay? What time you get up? 10, right? No tuition in the morning, right? How about you? Yeah. Maybe when school starts, later they'll start taking extra class in the morning. But not, you don't have extra classes, right? Yeah, that's it. Because they teach. Here they don't teach. So they every Saturday they call them. Yes. So if there is any extra class, we'll move it. Okay? Right. So see you tomorrow, 10 o'clock. Yes. Revise this. I'll send you the presentation and recording also. Okay? Yes. If you want, you can watch that later. Yes.